Hello and welcome back to Science of Medicine. Today I want to follow up and talk about the hematologic system. You may remember that when we were talking about the musculoskeletal system, I mentioned that one of the functions that bones serve is as a reservoir for making blood cells. Well, that's what we'll talk about extensively today. Now, the hematologic system has several components. First and foremost are the red blood cells, or erythrocytes. These are some biconcave disc-shaped cells that are pretty small. They're generally about 6 to 8 micrometers in diameter, although they can deform. They're formed in this trabecular portion of the long bones predominantly, but also in the pelvis and other areas. There's no nucleus. They're formed from precursor cells and they're expelled from the bone marrow. They are therefore limited in their lifetime. They have about a 3 to 4 month lifespan. They contain hemoglobin, which is the oxygen carrying molecule, which is essential for doing one of the functions of the erythrocytes to carry oxygen to the cells, and also carbonic anhydrase, which will facilitate the conversion of carbon dioxide into bicarbonate so that it can be transported back to the lungs and then offloaded as carbon dioxide waste. The hematologic system is also comprised of the spleen, which is responsible for the destruction of those red blood cells after they've served their utility. Anytime you have a fluidic system, you're going to have leaks. And in the blood vessels, there are inevitably going to be ruptures and leaks. And so you have to have a way of actually dealing with this. There are platelets, which are also formed in the bone marrow, but last considerably less time, generally about 10 days, that are essential in the blood clotting cascade, which we'll talk about later. And then also a sequence of proteins that are also necessary for blood clotting, but more about that later. So let's talk about erythrocytes. So the red blood cell is composed of 4 proteins, or globin chains. And each of these chains contains a porphyrin heme molecule, which is shown here. At the center of this porphyrin is an iron 2, and that iron is going to be the moiety that then binds to molecular oxygen and allows for it to be carried to the cells. Now, if you can't make porphyrin with iron, then that leads to a deficiency in the red blood cells, which is called an iron deficiency anemia. Because the cells themselves are structurally smaller than normal erythrocytes, this is called a microcytic, for small cell, anemia. Now, there are also other things that are necessary for the synthesis of the erythrocytes, and those include certain cofactors in enzymatic reactions, such as vitamin B12 and folate. And if there's a deficiency of these, it leads to a macrocytic or large cell anemia. We'll talk about that more in just a second. Now, there are some slight variations of the hemoglobin chains, alpha and beta, gamma, delta, and we'll talk about these when we talk about hemoglobinopathies. So how do I make my erythrocytes in the bone marrow? So most of the porphyrin synthesis occurs in mitochondria, and that's because a lot of our iron is sequestered in there. If I have iron floating around in my bloodstream, a lot of bacteria will use this, and so that can't be the case. So I have a circulating protein that binds very strongly to iron called transferrin, which will transport it to mitochondria, and then that'll allow for the synthesis of using iron. The globin chains are synthesized on ribosomes that require both vitamin B12 and folate for the DNA processing. And if you remember, I mentioned that there was a macrocytic anemia that could result from deficiencies of either of these, although in the United States, vitamin B12 is much more common as a deficiency than folate. That leads to a macrocytic anemia. The red blood cell is pretty simple. It's actually composed of a lipid bilayer with some structural proteins that allow for it to have this biconcave disc shape that I showed you on one of the earlier slides. It's about 6 to 8 microns in diameter, but it can deform to pass through the smallest of capillaries, which are generally about 4 micrometers. The mature erythrocyte contains hemoglobin, the plasma membrane, the cytosol with those proteins, a sodium-potassium ATPase pump, but no nucleus. There's no need to make anything else, which means that as things break down, such as structural proteins, the biconcave disc becomes a sphere, and then that can be destroyed in the spleen. And that normally takes about 120 days, although there are many reasons that it is actually less than that. So if I look at a structural drawing of a hemoglobin molecule, I see something which is comprised of these tetramers, the 2 beta units in red and the 2 alpha units in blue, as well as the 4 porphyrins, which are also shown in here that will carry the molecular oxygen. And then this hemoglobin will be present in my red blood cells to be able to carry the molecular oxygen. Now, as I mentioned, since these erythrocytes don't contain a nucleus, there's no creating any kind of new proteins. Everything is synthesized and packaged into this. So eventually, my structural proteins that keep that biconcave disc will actually go away. They'll degrade, and it will become a sphere. And the spheres of hemoglobin, of erythrocytes, are going to be destroyed in the spleen. Now, the spleen is located in the left-hand portion, upper left-hand portion in your abdomen, and it has areas that are generally reddish, that is called red pulp, and some areas that are in the center of this that are called the white pulp, which are involved in your immune system, and we'll talk about that the next time. Now, in this red pulp area, there are these structures that are almost like the slats of a barrel with spaces in between that allow for biconcave discs to actually pass through, but spheres basically bash up against that and are degraded. When that happens, the degradation products then are broken down eventually into amino acids and a yellow compound called bilirubin. Bilirubin we can see in our fecal matter. It turns things somewhat yellowish brown. And also sometimes when people have what's called a hemolytic anemia, where red blood cells break apart much more quickly, you can see a person will have jaundice or yellowing of their whites of their eyes, the sclera of their eyes, or of their skin or other areas. So I mentioned that this destruction of erythrocytes occurs in the spleen, and it produces bilirubin, which is a breakdown product of hemoglobin. And I mentioned to you also about these hemolytic anemias where you break down. And this young, cute little child right here actually has jaundiced eyes. You can see the sclera, the whites of the eyes, are actually yellowish. So, if I have erythrocytes in my hematopoietic system, sometimes I have too few of them. I do have some conditions where I have too much in the way of red blood cells. There are certain malignancies that do that, but the lion's share of hemoglobinopathies are when I will have too little. And you can think about these too little erythrocytes as comprising a set of diseases, conditions which are referred to generically as anemias. And those anemias can result from a couple different sources. Number one, I can form too few erythrocytes. I don't form the red blood cells enough, and therefore I have too little. Number two, I could destroy the red blood cells faster than I'm making them, and those are the hemolytic anemias. I could form erythrocytes, but they're abnormal, and that leads to their premature destruction. Or I could have a fluidic leak and simply bleed the erythrocytes out. So let's talk about them in detail. Too few erythrocytes that are formed. That is, I'm not making the red blood cells fast enough. And that can occur because there's a problem in the bone marrow with the cells that make the erythrocytes. That could be something infiltrating the bone marrow, cancer. There are other things that are there. So-called aplastic anemias, which is where you have some chemical that's toxic to those cells that make the red blood cells. There are a number of medications and also toxins that do this. I could have a problem with not having enough of the starting materials to be able to synthesize the porphyrin or the erythrocytes. For example, iron, leading to the microcytic anemia, or vitamin B12 or folate, which leads to the macrocytic anemias. I also need a hormone called erythropoietin. Erythropoietin is actually synthesized in the kidneys, and if I don't have enough erythropoietin, then I can't give the stimulatory effects to the cells in the bone marrow to make the hemoglobin and make the erythrocytes. All of those lead to too few red blood cells being made. I can destroy the red blood cells fast. Those are hemolytic anemias. And the main class of that are when I have problems with the membrane. So I have that biconcave disc shape. If that biconcave disc shape is not present, then I will destroy them in the spleen. Or if I have things that cause the faster destruction of the red blood cells in the spleen, I will have a hemolytic anemia. For example, there's a hereditary condition called hereditary spherocytosis. You inherit this. And it's a defect in one of the proteins that actually makes them into the biconcave disc. And you end up making spherical red blood cells. Those are destroyed then in the spleen. There's a deficiency in an enzyme called glucose 6-phosphate dehydrogenase. It's hereditary also. And under certain conditions, such as oxidative stress from illnesses, sulfa drugs like Bactrim, which is used for a lot of things like urinary tract infections or some skin infections, or even fava beans, can render the erythrocyte more likely to be damaged from this stress and therefore break apart. There are also anemias, hemolytic anemias, where the body will attack itself. It will attach antibodies, for example, to the red blood cell inappropriately, and that will lead to their premature destruction in the spleen. There are some genetic hemoglobin disorders, such as sickle cell disease or thalassemia, that results in the formation of red blood cells that are more likely to be destroyed, and we'll talk about those in a few minutes. And as I mentioned before, you could actually have a leak in the system causing blood loss directly. So let's talk a little bit more about genetic disorders. There are a couple that I'll talk about. There are many hemoglobinopathies like this. One of them are the classes of what are called thalassemias, and that's a problem with alpha and beta chains, and there are different combinations of this that basically lead to a reduced rate of synthesis of one or more of those globin chains, and therefore an anemia. Sickle cell disease is by far a very prominent disease that is present, and hopefully off into the future will be one that we'll be able to correct. Sickle cell disease, there's a single point mutation in a particular amino acid that leads to, under conditions of low oxygen, a sickling of the cell, a deformity that makes it more likely for the erythrocytes to polymerize and therefore make it so that they get stuck in the capillaries, and then anything downstream of that flow has hypoxemia or is not able to get erythrocytes and therefore oxygen, leading to very painful sickle cell crises. Now, one of the things that has happened in the last few years is gene editing technology called CRISPR, and there is currently a clinical study that's undergoing that is tried to correct for this sickle cell disease. In fact, it was reported on very recently in the New England Journal of Medicine about some of the early success for this gene editing technology in treating sickle cell disease. Now, if I look at red blood cells and I characterize them, I find that those red blood cells come in different types. And we characterize this as a blood type. And effectively, if you look at the surface of the red blood cells, they contain certain molecules. And those molecules might interact with things that can be in the blood serum. The things on the red blood cell are called antigens, and the things in the blood serum that might react with them are called antibodies. And that can lead to premature destruction of the erythrocyte, process called agglutination. And we discovered many, many decades ago about compatibility. There are three main antigens, if you will, or antigenetic categories, the ABO comprising this. And these are determined by alleles that you inherit, one from your mom and one from your dad, that encode for A, B, or O. And there are 6 combinations that lead to types of blood. There's the allele O with another O that leads to blood type O. There's blood type A, which could either be alleles AA or AO. There's blood type B, where you have alleles BB or BO. And then there's blood type AB, where you have alleles A and B. There's also a rhesus factor, or RH factor. There are different types. There's 6 common types, but the D antigen is the most important. And they divvy up into 2 types. Those that contain the D antigen, which are called RH positive, and those that do not, which are RH negative. And let's investigate this a little bit further. So just so that you and I are on the same page, we can use very simple kinds of genetics to be able to look at likelihoods of things. Now, O is kind of the recessive trait in this. It will not be dominant when combined with A or B. So suppose I have a child and I'm looking at the likelihood of that child having AB blood type if mom is blood type A and dad has blood type AB. So I want you to take a moment and see if you can actually look at the Punnett squares to be able to determine what this likelihood is. So go ahead and pause this, and then when you're ready, click back on. So I hope that you recognize that since mom is blood type A, that means that she could either be AA or AO, and dad being AB can only be AB. So if I look at the Punnett squares for that, if mom is AA, dad is AB, I have these possibilities where 2 of the 4 combinations would be blood type AB. But if mom were AO, dad's AB, I have 1 of the 4 combinations, which would lead to AB, meaning that I would have 3 out of the 8, or 37.5% possibility of a child having an AB blood type, with mom being A and dad being AB. So let's look about this ABO system and link this to transfusions, because this is one of the ways that people actually can run into trouble. So if I look at my erythrocytes, all right, if I have blood type A, that means that I express on the surface of that red blood cell type A antigen. Now, it's not really an antigen. It's not trying to make something up or whatever. It serves a function. But people that have blood type A antigen on the surface can't have any antibodies that are A, otherwise they don't live, because those antibodies would basically just clump on the surface and you would destroy your erythrocytes and you wouldn't live. But they do develop antibodies to type B in the blood. So consequently, if a person with type A blood were given a blood that attacked this A, then it would actually hemolyze this. So we'll talk about that in a second. It would actually lead to a problem. Type B blood has B antigen on it, and therefore in the serum it will have anti-A. AB can't have in the serum any antibodies, but type O, which has no antigens on its surface, has both A and B antibodies in that. So what this means is that if you have any mixture of any 2 blood types, and the hemolysis, the transfused blood, is going to occur, if you happen to give blood that to the person that has an antibody in their serum to that blood. And what that means is that, for example, type O, of course, is not going to have any antigens on it, so anybody can get this. They're not going to actually have any type of hemolysis of the blood. But AB is going to be able to take any kind of blood because there are no antigens to react with with those erythrocytes. But A could receive blood type A or O. B could receive type B or O. Now, I mentioned to you before that anytime you have a fluidic system, you can have leaks in that process. And there's a way that we actually deal with leakage, and that is through this coagulation cascade. So we have blood vessels, and if there's a break in the blood vessel, you develop a leak. So you have to have a way of basically dealing with that leak and repairing it. On the short term, that's going to be dealt with by forming a blood clot. And that clot is going to be formed by making a protein fibrin, which is going to cross-link and have platelets sort of impaled on it to form a temporary barrier. Later, other cells will come in and actually repair the blood vessel and destroy the clot. But this is how you're going to do this. Well, why don't we just have fibrin floating around in our bloodstream? Well, the reason for that is if we did, it would clot everywhere. So we can't do that. So what we have is a protein, fibrinogen, which can be converted to fibrin under the actions of thrombin. So if I have fibrinogen around, which is made in the liver along with these other coagulation proteins, and thrombin is present, I will form fibrin, and then I will be able to actually form a clot. But if I had thrombin floating around, then I would have a problem with forming clot for the same reason that if I had fibrin floating around. So I actually have prothrombin floating around, and it's activated by factor 10A. So if 10A is present, prothrombin goes to thrombin, and then that will convert fibrinogen into fibrin and make my clot. But I can't have factor 10A going around, otherwise I would have clotting. So I have a sequence of cascades that actually lead to the formation of factor 10A. And they all start with the underside of the endothelial layer expressing a factor called tissue factor that will interact with platelets. So I have a cut or a defect in that such that I will activate tissue factor, and then that will cause a cascade that will eventually lead to factor 10A, which will then take my prothrombin to thrombin. Thrombin will take fibrinogen to fibrin. Fibrin will form my clot. Now, under most conditions, then, I will have a system that will clot only if I have some sort of rupture in my blood vessel. I also have a process to remove clots. There's a molecule called plasmin, which will go around and do this. And normally my rate of clotting and rate of removing clots is equal. But if I open up a blood vessel, then it will actually tip that balance to forming a clot. Now, the reason I go through and spend some time on this is that there are numerous anticoagulants that we have now to keep people from clotting. There are inhibitors of factor 10A, and there's one of thrombin also. We can also inhibit several of these factors here with the use of a drug called warfarin, and if that's the case, then it prolongs the prothrombin time because I have fewer of these factors. All right, let's talk about a question. So suppose a patient has hereditary spherocytosis and their whites of their eyes appear jaundiced or yellow. Why is this? Do they have increased vitamin B12 deficiency, which lowers their bilirubin? Do they destroy the red blood cells that leads to higher bilirubin? Do they have an iron deficiency, which leads to higher bilirubin production? Or do they synthesize globin at a lower rate, and therefore that lowers their bilirubin usage? I hope you recognize that hereditary spherocytosis leading to jaundice is one of those increased destruction processes that I spoke about in terms of anemia, and that leads to red blood cells being destroyed. I hope you have enjoyed this introduction to hematology and the hematologic system. When we talk next, we'll talk about the immune system, and I'll try to do this in a way that will help you understand how you fight off infections. As always, if you enjoy this, please feel free to subscribe so that when new videos go up, you're aware of that. And if you have any comments or like this, give me some feedback. Thank you, and I'll see you again soon.